Okay, so yeah, thank you for the opportunity to speak here today, and welcome to everybody who is listening in. So before we start, sort of a first few things. First about me in the lab. So I'm essentially a basic, so a fundamental scientist, and I've been working on this O-GIcNAc modification since 2005. So way before it was clear that this was linked to any kind of disease. Some of you will have maybe seen from my work or publications that we were in at the University of Dundee in Scotland, but since about 2 years now, me and the lab have moved to Aarhus University in Denmark. I should, I guess, emphasize in a different way that I'm a basic scientist, so I'm primarily driven by curiosity, and the way that I view the world and design experiments is basically it's completely evidence driven. The other thing that I think, like Lance, whose talk I saw, and there will be some sort of overlap between introduction, but he said, and I want to emphasize again myself, is that I am not a medical doctor, so I cannot possibly comment on individual cases or symptoms, or in particular treatments of the patients and their families around that. I can only see my own screen. However, I'm totally open to being interrupted and for you to ask questions. I think Andre will be able to see your questions and maybe can just ask them verbally, and I can answer them straight away. So the other thing I wanted to say before we start is language. There may be some of my scientific colleagues listening in. I should apologize to them that this talk is entirely pitched at a lay and not a scientific audience. So some of the data and controls and tests that we normally show, I'm not going to show them. I should also say that sometimes maybe as a scientist, some of the things that we say may sound abstract or maybe sometimes even harsh. Whereas obviously I'm aware that we are talking about human beings, children very often, who have this disease, and yeah, so there's sometimes maybe a little bit of a disconnect between my world as a basic scientist and the reality on the ground of patients with OGT-CDG, and that is obviously not intentional. It's also important to understand that sometimes scientists, medical doctors, and people working in the pharmaceutical industry, they think and work and communicate in very specific and sometimes seemingly quite odd ways, and that is because those people are aware that in the end there are much more unknowns than knowns, and therefore we might express ourselves in a somewhat caged way where we introduce or words that indicate uncertainty. Basically, we don't know a lot. Also, I would extremely strongly encourage you, if you are a layperson that is not a scientifically trained person, and you're overwhelmed by everything that you have heard and that you have read because you've become familiar with this disease, and now suddenly you have to think about science and medicine, read these two books, because they will tell you what are good ways and bad ways to do science, and good ways and bad ways to develop drugs, and just to make sure that we always stay on the right side of things. So I'm aware that maybe some of the people that are listening in, or actually most of the people, will be families with children with OGT-CDG and don't necessarily have a scientific training. So just to make sure that everybody's on the same page, and I know that Lance already did that, but I feel I have to do it again. Not because he didn't do a good job, he did an excellent job, but maybe there's new people listening in here. It's just extremely quick biology 101. So you all have heard of DNA. So DNA is the carrier of the genetic code in our bodies, in all cells of our bodies. It basically provides a blueprint of building an entire human being. Ultimately, however, the sequence of the basis of the DNA codes for proteins. So proteins are a linear chain of amino acids. They fold up to make a particular function. There are about 20,000 different proteins like that in every human cell. And so basically what happened around 2 billion years ago is that that there was the first replicating unit of life, probably some kind of bacterium. And in this bacterium, we have proteins that carry out completely different functions. So all of these different little black dots that you see in this cell, and this of course is magnified thousands of times, are proteins, and these proteins have specific functions. And that was fine. So life existed like this 2 billion years ago. And then at some stage, it became necessary for life to become multicellular. So that instead of just one cell, basically cells clumping together. And this is what we as human beings are. And you can see here the development of a vertebrate embryo, in this case of a salamander, from single cells dividing into complex folded structures that ultimately give rise to an embryo. And that is possible because cells now talk to each other. So they don't live like individuals anymore. They clump together to make an organism, which is what an example, of course, that we are. And when that happened, something became necessary. And that is, there was a need no longer to have just 20,000 different proteins that have these different functions to make cell, but also the cells needed to talk to each other, and they very often do that by small protein modifications. And I have highlighted some of these modifications here in different colors on the different proteins. And these protein modifications, you can think of them as switches. They can switch on and off the function of individual proteins. And what then happens is that we get a very, very complicated picture. There's no need to look at all the words in this graph, but these are all different abbreviations of proteins that can switch each other on and off. So what we get is like a circuit of molecular switches. And this is remarkably similar to a computer circuit, where we have different parts of a circuit that have different functions, and they get switched on and off. So essentially, by having these 20,000 different proteins in the cell and by having about 100 different types of modifications, we arrive at a way of achieving multicellular complexity and building things like us, human beings, with all our complexity. So we then arrive at what this O-GIcNAc is. So this O-GIcNAc is nothing else than one of these, say, 100 possible protein modifications, one of these switches that can control what a protein exactly does. And we know that this exists on about 5,000 different proteins in the cell. We don't really yet fully understand what the function of this is. Does it switch on the protein, or does it stop it being made, or to live in a different part of the cell? But we do know that this is pretty important, because the loss or reduction of this particular O-GIcNAc protein modification leads to intellectual disability in children. If you try to remove this modification from cells, they die. And for instance, even in other organisms like flies, if you take away this modification, you get very, very strange effects on the building of the body plan of these flies. So now I switch to the second part of my talk. This one over here.
Daan, that's actually exciting to see how you compared the human cells with the computer circuit.
Yeah, that is. Can you see the next slide, Andre?
Yeah, I just put it on the screen.
Great, excellent. So, so now a little bit more about this protein O-GIcNAc modification. So we can have proteins, they can be modified with this O-GIcNAc switch, which is shown here in purple. And this modification is actually carried out by another protein that is called O-GIcNAc transferase, and the modification can be removed by the protein is called O-GIcNAc hydrolase. And for the rest of my talk, we abbreviate these two proteins as OGT and OGA. So this modification, again, just to remind you, found in 5,000 proteins, is absolutely essential for life, but we don't understand really why. So we do know that it's essential for development. If you remove OGT from even embryonic stem cells, so the very, very early cells that form an embryo, that is lethal. I've already shown that if you remove OGT from flies, that leads to lethality, so death of the fly, but with very strange changes in its body plan. In fish, if we reduce levels of OGT, we get embryos that are truncated. And in mice, if we delete OGT or OGA, that leads to lethality around birth. But what has happened about 8 or 9 years ago is that both the lab of Lance Wells and my lab in Dundee reported the first missense variants, so that these are little errors on the DNA that make an error in the OGT protein that leads to a disease in humans. And this we published together in a review when we had collected about 7 different families, and what we saw in these families was that that leads to intellectual disability, developmental delay, a number of other symptoms that I will discuss later. And we called this O-GIcNAc transferase congenital disorder of glycosylation. Now, I just want to give you an example, and this is relevant because this is actually a family that will come back later in the talk. So this is what we call a pedigree. So all the squares are males, all the circles are females. The ones with a diagonal cross through them means that they're deceased. And the ones that are shaded in gray means that they carry some symptoms of the disease. And this is basically an example where it is the mother that has 2 copies of O-GIcNAc transferase, and it has transferred they've transferred one copy to one healthy son, which is the one born in 1960. It has a particular amino acid that we call cysteine, or CYS. Whereas the 3 other brothers are affected because they have a wrong amino acid at that place, because they have a little error at the DNA sequence that codes for this protein. And these are some of these brothers. The first one born in 1962, and in 1966 they were born as twins. And this family is actually pretty unique because these are pretty much our only examples of patients that are in this age category. Most of the patients that we've heard about are very, very young children. These men have intellectual disability, developmental delay, some autistic features. They have seizures, which is something that we find in many OGT-CDG patients. They have some facial dysmorphic features and some skeletal defects. But otherwise, they are able to live semi-independently and reach the age of well into their 60s. So what we currently know about these different variants of OGT-CDG is well beyond what we have published so far. So we in the lab have contact with clinicians and families of over 60 variants. I think as of today, it's actually 64. And that means that we can now look in much more detail at the different symptoms that we have found in the first group of patients and start to see also new things. So the most common symptom in this group of patients is intellectual disability. So you can see that in the section of the nervous system. Essentially, all of our patients have intellectual disability. Many of them, however, also have seizures. This was maybe not so apparent from the first group of patients. Also, many of them have heart malformations that we have just started to study. So by collecting more patients, we get more insight into what are the most commonly occurring symptoms. So if we look at the structure of this OGT protein, and you can represent that as a linear sequence of amino acids, as we discussed in the beginning. That's what a protein is. It's just a string of amino acids coupled together. Or we can look at the 3-dimensional shape, which is this funny thing that you see rotating in the back. We can see where all of these mutations are located, and they're basically located all the way across the protein. So the questions that we have as a lab, and there are actually many more, but I've highlighted some of the most pertinent ones here, is some of these. So, how do we know when a clinician contacts us with a mutation in the DNA that codes for the OGT protein? How do we know that that mutation really causes the disease that we see in the patient or the developmental delay? An absolutely critical question, and this is something my lab is very aggressively pursuing, is whether this is a neurodevelopmental or a neurodysfunctional disease. So these are two complicated words, so let me just explain this in a different way. Is it the case that in these young children with this disease, that basically there's something that goes wrong during development of the early embryo, and that is structurally and fundamentally deposited, and that we therefore cannot correct anymore? Or is it that actually the structure of the brain is normal? It's just that the neurons that are in the brain are not working properly, in which case it may be possible to correct some of the symptoms. So this is, I think, very, very relevant for this group of patients, because if it's purely a neurodevelopmental disease, it is unlikely we will ever be able to find any kind of cure. Whereas it's a purely neurodysfunctional disease, that means that we can somehow maybe in the future find ways to help neurons talk to each other properly, and therefore help cure this disease. What types of cells and circuits are affected in the brain? What proteins are affected? What O-GIcNAc proteins are affected? How does all of that lead to intellectual disability? Can we discover new drugs to treat this disease? And can we think about alleviating symptoms with inhibitors, food supplements, or even things like gene therapy that I've heard was discussed in some of the previous talks? Now, the crucial thing is, none of these questions we can really reliably answer without having animal models of OGT-CDG. So why is that? Well, let's start with what we know. We know that there are young patients with variants in the OGT gene that leads to this OGT-CDG symptom. We can do very, very little with these patients to answer some of these questions. We cannot look or take samples from their brains. We cannot do all kinds of treatments with them. There are huge ethical concerns associated with that. Basically, we as basic scientists certainly don't do that. So we need to find a way of learning more about this disease by transplanting this disease into other organisms. And that is exactly what we have done using this new technique that you may have heard of, which is called CRISPR. So with CRISPR, we can basically engineer the DNA in other organisms so that we have exactly a copy of the same mutation that patients have into the DNA of that organism. So in that way, in the lab, we have made stem cells, which are cells of a very early embryo, and also in this case of mice. And we can see that when we introduce these patient mutations using this CRISPR technique, that leads to effects on making neurons from these stem cells. We've introduced it in flies. This is an extremely useful model organism for, in particular, when it comes to screening for drugs or drug development programs, where if we introduce some of the patient mutations that we know, we see effects on sleep of those flies. And we also, when we go and look at the neurons, we can see clear neuronal defects. But probably the most relevant for today's talks is mice. So in mice, we have now succeeded for 3 different patient variants. And I know that some of the people that are listening today, basically it's their children whose mutations we have introduced in these mice to study this OGT-CDG disease. So I'm not going to show a lot of data because that's really not what this talk is about, but I just want to share with you, and some of these are already published and some of them are not, that basically when we introduce these patient mutations and we look at the brains of these mice, they do have reduced O-GIcNAc. So that is something that basically is copied in the mice and in humans. What we also see is that these mice are smaller. They have lower weight, and they have a very strange change in their fat mass that I won't discuss further. They show hyperactivity, They have microcephaly, so that means that they have a slightly smaller head and a brain. They have locomotor defects, or maybe in sort of common speak, you could say that they are somewhat clumsy. And they also have unexpected changes in the structure of their neurons. And I just want to give you a little bit of a snapshot of some of the data. So basically, what we are looking at here is a normal, healthy mouse that doesn't have any mutations on the left. And on the right, we have a mouse that carries one of these OGT-CDG mutations. And what we do is we put these mice in a box and we ask them, or we give them an opportunity to explore the box. What you can see on the left is that this normal, healthy mouse, it goes around the box, looks around, looks at its environment, whereas the mouse that carries the OGT-CDG mutation basically walks around in circles without maybe knowing where it wants to go, why it wants to go there, what there is to explore. Maybe it doesn't even remember where it's been before. Basically, it looks like either sort of a hyperactive mouse or a mouse with a memory defect. So we can capture that a little bit more quantitatively, if my slides want to continue, in graphs like this, where we can just look at the entire track. So here you can see it just with your bare eyes, that basically these OGT-CDG mice, and in this case, it's the same mutation as I showed from the family that I showed the pedigree of earlier. Basically, they are hyperactive. They go all around the box with no rhyme or reason, whereas the wild type or the normal mice, the non-mutated mice, behave in a completely different way. We can also look at the skulls of these mice, and these pictures on the left, they kind of indicate the differences in the skulls. So where maybe the wild type skulls are larger than the mutant mice. And basically, to summarize this, we can then do measurements on these skulls, and we can see that the skull of these mice are slightly lower and they're slightly shorter at the back. And this is where the cerebellum is located, which is a particular structure of the brain that is in particular relevant for motor skills. And we can see in some of our patients that the motor skills are affected. And finally, when we then take the brains of these mice and we compare unmodified mice to mice that modified with these OGT-CDG variants, we can see that there are gross structural defects in these neurons. Basically, you can see this fuzz. They look more like a tree, and this could indicate a hyperconnectivity and maybe explain some of the hyperactivity that we see in these mice and also in our OGT-CDG patients. So then, kind of summarizing this, what we can see in these mice is that we seem to have reproduced some of the features that we see in patients with OGT-CDG mutations in the mice carrying exactly the same mutations. So we can see that they're smaller, they have a different shape of the skull, so dysmorphia in the face or microcephaly. They clearly have developmental delay. They're very small when they're born. They have a degree of clumsiness, so psychomotor retardation, and they maybe have a degree of hyperactivity, which maybe matches onto this idea of them being hyperactive or having ADHD. So what will we do next with these mice? So the most important question to me is to answer this question, which is whether the defects that we see in these mice, are they because of something that happens very early on in development of the embryo and therefore we can't correct after these mice are born? Or is it that there's a problem with the nervous system of these mice that doesn't work properly, but is properly in place and that we can try to correct? You know, what types of cells or circuits are affected in the brain, what proteins or O-GIcNAc proteins are affected, how does all of that lead to intellectual disability, and then this question of whether we can alleviate symptoms with inhibitors, food supplements, or gene therapy. I know that some of you already have children that are on food supplements. I personally believe it is really, really important to first properly test this in mice before moving on to any kind of a treatment, but that is a choice that you have to make for yourself and your children. So some of the challenges of thinking of maybe a drug, so getting a pharmaceutical drug to treat this disease, one of the absolute key questions here is what is the size of the patient population? And this is difficult because with other genetic diseases, such as, for instance, sickle cell anemia or cystic fibrosis that you may have heard of, that is a type of mutation that is very widely spread through the population, and therefore you can calculate very accurately how many people per year throughout the world are affected. This is not possible here because most of these mutations are de novo. That means that neither mom or dad carry this mutation. It occurs at the time of conception. And to calculate the frequency of that happening is, and I've talked to many colleagues, is nearly impossible. But this is an absolutely crucial parameter for, say, attracting funding or getting a pharmaceutical company interested. If basically it is 1 in 10,000 live births that have a mutation in OGT and that leads to OGT-CDG, that means that it's a very significant patient population that many pharmaceutical companies would still be interested in. If it's only 1 in a million, that means it's an extremely rare disease, and it will be very, very unlikely to get companies interested in investing in such a program. Another sort of advantage is also at the same time a disadvantage is that actually there are already many companies, about 4 or 5, that are developing inhibitors for OGA, which would be a very obvious strategy for maybe treating OGT-CDG. But they are targeted towards neurodegenerative diseases, so Alzheimer's and Parkinson's disease. And I've actually contacted many of them, and they're not interested in using their molecules for OGT-CDG because they are focused on these neurodegenerative diseases. So we could, think about a new or our own program. And we have, of course, many of the tools. We are, as far as I'm aware, the only lab in the world at the moment that has a mouse model of disease, but we don't have the millions required to do this or the pharmaceutical partners that would be interested in helping us. And the other thing to keep in mind is that the timing of this type of route would be 10 to 15 years. And I know that's quite disheartening maybe to some of the patients and their families listening in, but that is the reality of a drug development program, that it takes about 10 to 15 years from an idea to get a drug properly through clinical trials and then actually be made available through clinicians. So finally, I just wanted to say that it's just me talking here, but I'm definitely not on my own. We have moved in Aarhus to a new and a great place to work. These are some of the young scientists in the lab that are working day and sometimes night to learn more about this disease. One of the reasons for coming to Denmark is that both the Vilum Foundation and the Novo Nordisk Foundation have given us very generous funding to basically just focus on the science and not worry about attracting funding to do these experiments. And there are many local collaborators and also international collaborators, and I particularly want to thank the clinicians, the patients, and also the families like you listening in for being willing to engage with us. And that's the end of my talk. Thank you very much. If you have any questions, please ask them.